Hi, everyone. I'm Mike Curran. I'm here to talk to you about some work our lab's been doing around hypoxia and how it creates a major barrier to the success of, spontaneous T-cell immunity as well as immunotherapy, particularly in what we refer to as immune cold tumors, those that lack prior immune infiltration, don't respond well to immunotherapy, things like pancreatic cancer, prostate cancer, glioblastoma. I'd like to thank the LabRoots team for inviting me to give this presentation as part of the immuno-oncology virtual conference here. So without further ado, I have some disclosures. Consider them now disclosed. And let's sort of set the stage with immunotherapy. So, what's evolved over the last decade is immunotherapy has become a real pillar of oncology, how we treat cancer patients. And, largely this is some work that I did a long time ago with this gentleman here, Jim Allison, winner of the Nobel Prize for sort of being a pioneer of this field of checkpoint blockade and immunotherapy, but really showing this is why immunotherapy has been so compelling, right? We can block individual immune checkpoints, markers on T cells that are engaged by tumors to shut off your adaptive immune response when it comes knocking, tries to eliminate the cancer. When they're blocked, now your T cells are not shut off and tumors are cured, even in very difficult to treat intransigent tumor models. This is a mouse model of melanoma that's extraordinarily difficult to treat. Combinations of these, so blocking these two primary immune checkpoints, are again switches that are engaged on T cells to turn off their function and shield tumors. CTLA-4 and PD-1 together, we showed, this could cure a very high percentage of animals and really shutting down this whole pathway. So blocking the interaction of these PD ligands with PD-1, as well as this B7-1 PD-L1 co-inhibitory interaction described by Arlene Sharp and Gordon Freeman, allowed us to cure the majority of animals. And, this was very successful in a mouse melanoma model. Often we see things successful in mice that don't turn out to be quite so successful in patients, unfortunately. This was. So this was actually, we were very happy to see that when you went into melanoma, which we now consider, a great success for immunotherapy, but a 2-year survival of about 80%, 60% objective response rate, and really, maybe 10 to 20% of these patients are durably cured of detectable disease. Just to put it in perspective, because, in the 2020s world, we think of melanoma as an easy cancer to treat with immunotherapy. In 2010, when this work started, the 2-year survival rate for melanoma was only 18%. So this has really been transformational in that setting, led to FDA approval of this as the first immune combination for checkpoint antibodies, and one that's now been extended to many indications. However, despite all that success in more immunogenic tumors like melanoma and lung cancer, what we call hot cancers, hot because they're immune infiltrated at baseline, they have a lot of mutations that are immune visible. Unfortunately, those cancers are a minority of all cancer patients, and the majority of cancer patients have these cold tumors. Again, cold tumors meaning they're not immune infiltrated at baseline, they have less mutations, neoantigen generating mutations for your immune system to target. And so even within, though, the patients that we can treat, which is the sort of 45% of patients with warm or hot tumors, the actual objective response rates are not as high as we would like them to be by far. So really under 15% of those patients that we actually treat, if we consider sort of the warm tumor patients as well as the hot tumor patients, actually have objective clinical responses, PRs, CRs. So really, there's a lot for us to understand about why some patients aren't responding, and particularly these cold tumor patients that make up the majority of cancer patients. We'd like to find a way to reach them with immunotherapy. And so even in the hot tumor setting, melanoma, where maybe we have a 60% response rate, that still means 40% of patients aren't responding. And so Roger Lowe's group looked into what are the origins of that lack of response in that 40% of patients. And one of the key gene sets they found enriched was hypoxia. So, disordered vasculature, lack of oxygen supply, oxygen-deprived tissue led to resistance to PD-1 blockade immunotherapy. Now, we were interested because we're interested in these cold tumors. I mentioned prostate, pancreatic. across mouse models, both transplantable and spontaneous, we found in the green here quite prevalent hypoxia. And if we look at published data from patients, we know that tumors like pancreatic cancer and prostate cancer are among the most hypoxic tumors in man. Also, for literally decades, we've known that tumor hypoxia in the clinic inevitably correlates with a worse outcome. And so what we found, and we weren't the only ones. I want to mention others such as Steve Hatfield and Misha Zikovsky's group, as well as Greg Delgoff's group. We all observed this, which is, this is our work in TRAMP-C2 prostate cancer under normoxia on the left. We see, there are T cells. The blue are CD4s, the red are CD8s. This T cell response is controlled by the presence of the white FOXP3 positive T cells. But that's a situation that we can reverse with current sort of checkpoint blockade antibodies. We could expand those effectors, suppress those Tregs, turn that into an effective tumor regressing response. The deeper problem, though, is in the right panel where you see this large area in green of hypoxia, and there are just no T cells there. No T cells there is a major problem for PD-1 antibodies, CTLA-4 antibodies, any kind of checkpoint blockade, because their substrate is essentially T cells. They are resurrecting exhausted T cell responses. They are removing engagement of these checkpoints on T cells so that they can't be shut off. But if there's none there to begin with, there's nothing for them to work with. And what you see on the right side is again, Zikovsky's data showing the same thing in a lung tumor. The green is hypoxia. The red cells are T cells. There are T cells there, but they're not in these hypoxic zones. So, so the problem then is why are we not seeing T cells under hypoxia? And the truth is, while we think about these checkpoint barriers, what we also need to consider are the metabolic barriers. The tumor is a very harsh metabolic landscape for T cells for a number of reasons. Hypoxia, which I mentioned, so lack of oxygen, T cells don't function well there, but hypoxia, fosters development of suppressive myeloid cells as well as immune suppressive polarization of cancer-associated fibroblasts. So T cells are suffering, their sort of arch enemies are flourishing. Also, nutrient deprivation. So a lot of these MDSC that are generated, other myeloid cells, such as tumor-associated macrophages, they'll deplete arginase, they'll deplete tryptophan, they release toxic metabolites, they deplete glucose and glutamine and other things that T cells need for sort of essential energy metabolism, as do tumor cells. So they're starved of the things they need on multiple fronts to actually carry out sort of successful metabolic expansion and anti-tumor effector function. T cells hate acid pH, and of course, within tumors, we have lactic acid deposition as a byproduct of Warburg effect, as well as, driven by hypoxia, carbonic anhydrase 9 upregulation and carbonic acid deposition. You can find, pHs in the tumor microenvironment well below 6.5, at which point, T cells basically just die. Adenosine release, another toxic metabolite, well, not really toxic, but suppressive metabolite for T cells, released from tumor cells as well as from other cells in the microenvironment that upregulate CD39 and CD73 extracellular ATPases that generate extracellular adenosine from ATP. And so why are we interested in this from an immunoregulatory standpoint? Well, if you're a T cell, this doesn't look good. I mean, there's so many levels of inhibition that you're going to face in this microenvironment. And if we actually look, why are we interested in hypoxia specifically? Many of these are actually in some ways downstream of hypoxia. So nutrient deprivation is worsened by hypoxia nucleating production of more M2 macrophages, more MDSC that are producing the arginase and the IDO. They're increasing tumor metabolism, so thereby, tumor metabolic adaptation to hypoxia leads them to deplete more glucose, glutamine, et cetera. Acidification is worse. You have more lactate deposition as well as carbonic anhydrase 9 being upregulated as a direct target of hypoxia-inducible factors. And then, adenosine release is also triggered by hypoxia. CD39 and CD73 are also HIF targets directly. And so all of this metabolic hostility ties back to hypoxia. So we wondered if we can find a way to antagonize hypoxia, maybe we can relieve some of this sort of suffocating pressure on T cell responses that occurs in these tumors, both directly and indirectly, at the level of vascular conditioning. I mentioned sort of nucleation of suppressive myeloid cells, really many levels of suppression such that T cells basically become non-functional and the environment becomes not survivable for an active T cell. So that led to a bigger question. How do you go about reducing hypoxia in a solid tumor? Now, if it's lung cancer, you can hyperoxygenate the lungs. But for any other solid tumor site, you're limited by the ability of heme and blood to carry oxygen, and obviously disrupted vasculature causes hypoxia. So, how do you address that problem? Well, we were fortunate in that, somewhat serendipitously, we recognized that there was a drug that was designed to be a hypoxia-activated prodrug. So much like a chemotherapeutic drug that would only be activated under hypoxia, TH302, that, in the early data, we realized there were signs that 302 was actually not working exactly as intended, but was having a really beneficial effect in reducing tumor hypoxia. And you can see that in the upper left panel here. So these are sections through whole TRAMP-C2 prostate tumors. The green is hypoxia. You notice there's very prevalent hypoxia in TRAMP in the untreated tumors. Checkpoint blockade doesn't really alleviate hypoxia, but the drug alone or in combination with immunotherapy is a remarkable reducer of tumor hypoxia, with 40% or so of the tumors being hypoxic at baseline, 5% after drug treatment. So really profound. In pancreatic cancer in the lower panel, which is really one of the most deeply hypoxic of tumors due to both abnormal angiogenesis and vascular stiffness and contraction due to the desmoplasia, we also see very effective reduction of hypoxia over time. Now, this drug doesn't reduce hypoxia immediately. Mechanistically, the way that 302 reduces hypoxia is it kills a selection of cells in this microenvironment, thereby initiating a tissue remodeling process that rebuilds the faulty, disordered vasculature into new, healthy vessels, really driven by sort of replacement of the abnormal angiogenesis driving, granulocytes, cancer-associated fibroblasts, and tumor cells with more of an M0 tissue macrophage-dominated population. And you can see that on the right, where we see an increase in CD31 positive vessel density with TH302 treatment. So great. So we can use this drug and reduce hypoxia. Does it actually improve tumor immunity? And what we see in the TRAMP-C2 transplantable models, these are tumors growing on the flank, these tumors actually are pretty sensitive to checkpoint blockade at baseline. You see in the red that about 50% of these mice can be cured with the combination of CTLA-4 and PD-1 blockade. Remarkably, we found that TH302 alone could actually trigger an immune-based reaction that would cure about 30% of these animals by itself. And that actually, is fostered by breaking down hypoxia and restoring T cell infiltration, leading to actually spontaneous regression, again, of about a third of these tumors. We know that's immune dependent because we repeated that experiment in RAG knockout mice, I didn't put the data here, that have no adaptive immune system, and the drug has no effect at all as a monotherapy. Now, what we do see here that's encouraging, though, both in the left panel and in the right panel, is that when we use TH302 with checkpoint blockade, they're very, very supportive of one another. So, on the left, about 85% of these animals are cured. On the right, about 95%. But basically, almost every animal can be cured of these tumors by the combination of immune checkpoint blockade and hypoxia reduction. The point of the right panel is to show that the closest chemotherapy drug to TH302 that is not hypoxia targeted, which is ifosfamide, doesn't have any of these properties, right? This is a sort of unique emergent property of very selective remodeling of certain cell populations only under the hypoxic regions of tumors. So if we look a little deeper, And we look into these prostate tumors and ask what's going on, both in the normoxic zones versus the hypoxic zones of the same tumors. Again, in the untreated state, and this time again, hypoxia is in green, T cells are in white, vessels are in red, the myeloid components in blue. What you can see is that there are very few white dots, but a few in the normoxic zones of untreated prostate tumors, none under hypoxia. When we use the drug alone, you can see this normalization of tumor vasculature, as well as where the yellow arrows are. You could actually see T cells now able to extravasate off of these newer, fuller, thicker vessels into these hypoxic areas as they break down. Now, checkpoint blockade, the combination of CTLA-4 and PD-1, is extremely effective at inducing enhanced T cell infiltration in the normoxic areas. But even with nearly 100 T cells per field in the normoxic image, you notice there's one or two, and that's it, breaking through into these hypoxic zones. This burden of suppression and really sort of hostility metabolically in these hypoxic areas is almost impenetrable by sheer numbers. So numbers alone of T cells will not overcome this. However, what you see when we combine TH302 with checkpoint blockade, we get a tremendous infiltration of the normoxic zones, and now you also see very high level T cell infiltration of these hypoxic zones as they're breaking down. The T cells can now survive there, they can now enter there, vasculature has been normalized, oxygen's returning, and that was something we saw very reproducibly, and it's key to the therapeutic benefit of this combination. Now, although T cells really dislike hypoxic areas, I mentioned, on the other hand, suppressive myeloid stroma tumors are nucleated there. They love it, they form there, it favors differentiation of M2 macrophages and myeloid-derived suppressor cells. A lot of this work was done by Dmitry Gabrilovich. And what you can see here is basically a reflection of that. You see that the overview of the GR1, the overlay of the GR1 and CD11B populations, the white and the blue, is very dense in the untreated tumors. There are lots of myeloid-derived suppressor cells. There are also lots of M2 macrophages overlaying the red and the blue. When we treat with the combination of immunotherapy and hypoxia reduction, you see how depleted that GR1 positive population is. This really suppressive MDSC population thins out tremendously, and what takes over is that remains. And what I'm not showing here is there's a phenotypic switch of those, not so much from an M2 fully suppressive phenotype to immunostimulatory M1, but just back to an M0 tissue macrophage, no CD163, no CD206, no arginase production, and really good at laying down new healthy vessels. An M0 macrophage would be something you'd think of naturally as being involved in wound repair. Now, if we look across the whole tumor, the impact of this on the MDSC population is pretty striking. Across the whole tumor, the frequency of arginase positive, so the really, verifiably suppressive MDSC drops by about 50%. And, if we look at it a different way in terms of density, we also see about a very substantial, maybe 50% reduction. So really what we're seeing is that, the establishment of this highly suppressive myeloid barrier throughout the tumor is pretty dependent on the presence of these hypoxic areas to act as centers for suppressive differentiation of these cells. So not only are these cells less prevalent, we also wanted to look at them functionally. And, in the untreated tumors, maybe a fifth of these granulocytic MDSC or PMN type MDSC are expanding, so they're Ki67 positive. We actually see a reduction in that proliferation with hypoxia reduction and immunotherapy. Now maybe only 5 or 10% of them are actually proliferating. So their numbers have been dramatically reduced, and the ones that remain are considerably less proliferative and able to re-expand and fill that gap. More importantly, when we take these cells out of the mouse and ask, how well do they suppress T cells, given there are very few still remaining, what we find is that if we look at a situation where we've CFSE labeled T cells, so they're fluorescently positive, as they proliferate, they dilute CFSE, so lower signal means more proliferation. With no MDSC, we see that the T cells have proliferated, diluted CFSE. Untreated, you see that they haven't proliferated. We see an intermediate signal with the combination of checkpoint blockade and hypoxia reduction. again showing that even though these MDSCs are still to some degree capable of repressing T cell responses, they've actually lost a substantial portion of their functionality in that regard. So there's less of them, they're less proliferative, and they're less T cell suppressive. That's actually kind of a major blow to the immune privilege that tumors rely on to exclude tumor immunity. Now, in some other experiments I'll tell you about, we saw hints that this response was actually more durable after we pulled the drug away than you would expect. Particularly, not so much in the transplantable TRAMP model, but in the TRAMP transgenic mice, where we wouldn't treat them for 3 months, and then we'd actually still see that the prior treated ones had a benefit in their microenvironment. So we wanted to ask whether when you take away hypoxia from these tumors, particularly when you do it in the context of inducing new immune infiltration, do you really durably compromise the ability of the tumor to maintain this immune privileged state? So the way that we did that, we established pretty large TRAMP-C2 tumors, so we let them establish for over a month before we started treating them. We only treated them with 1 cycle of TH302 with or without CTLA-4 and PD-1 antibody, waited a couple days, and then we transferred into those mice congenically marked bone marrow progenitors, asking the question, have we compromised the ability of these tumors to then attract these new congenically marked bone marrow progenitors and turn them into new MDSC? And so we waited a couple days, checked again, and what you see in panel A is that the combination treated animals, those that received hypoxia reduction and checkpoint blockade, had a dramatically reduced frequency of granulocytic MDSC. So things that looked phenotypically like granulocytic MDSC drawn from these new progenitors were significantly reduced relative to the other groups. But when I say things that looked phenotypically like granulocytic MDSC, the interesting thing is when we looked functionally, so in panel B, you see that unlike sort of a card-carrying MDSC, the ones forming in the mice that had this hypoxic region reduced, they didn't really successfully upregulate arginase, a critical enzyme involved in T cell suppression, so really one of the main drivers of MDSC-mediated T cell suppression, and they didn't downregulate fully MHC2. So as you're moving, let's say, in the myeloid spectrum from M1 to M2, the critical shift is you're upregulating suppressive functions like arginase, you're making more TGF-beta, you're making more PD ligands, and you're downregulating immunostimulatory functions, less CD86, less MHC2. So you see that although phenotypically, in terms of markers, these might look like MDSCs, functionally they haven't made it all the way there. So this really did tell us that hypoxia reduction compromises functionally these cells quite profoundly. All right, so all of this was really interesting, but everything I just showed you is in a transplantable model of prostate cancer that I mentioned at the beginning is pretty sensitive at the baseline to immune checkpoint blockade. It does respond to the combination of CTLA-4 and PD-1 blockade pretty well, responds a lot better when we also diminish hypoxia, but the whole driver for doing this research is that patients with prostate cancer, particularly castrate-resistant prostate cancer, don't respond to checkpoint blockade. And so we wanted to look at a model that similarly was non-responsive to checkpoint blockade. And so TRAMP mice, so these are spontaneous tumor-developing mice. They start expressing large T antigen in their prostate. By about 16 weeks of age, that means they spontaneously develop carcinoma in situ as well as local metastatic disease that progresses until, by about 36 weeks of age, they're starting to die. So we waited till that pretty late time point of 16 weeks to start treating them. We treated them for about a month with either TH302 alone or the combination with checkpoint blockade. And then we followed them for about 3 more months with no more treatment. At 36 weeks of age, we euthanized them and we looked at the weight of their prostates. Now, one other caveat about TRAMP mice that's worth mentioning, they get about 80% adenocarcinomas and about 20% neuroendocrine prostate tumors. So that is one thing to be aware of, is it's reflected in the data. So what you can see in the first panel on the right, CTLA-4 PD-1 blockade doesn't help these animals, right? They have as much tumor burden, their prostates are as heavy as untreated animals. Now, TH302 alone, you have a slight reduction in the size of the adenocarcinomas, which are the circles. no benefit to the triangles, which is the neuroendocrine tumors, and very little benefit overall, although some. What we really see that's striking, though, is if we combine checkpoint blockade that alone is completely ineffective with TH302 that has at best a marginal benefit, we get a dramatic reduction in prostate weight with, remarkably about a third of these mice having the same sort of weight of prostate as an age-matched normal control mouse, despite being genetically programmed to develop these cancers, constantly throughout their life. So we're initiating an immune response that is constantly active in controlling emergence and expansion of new cancer in these mice. The other thing that was really striking, and actually, if you're wondering the difference, the panel on the right shows, both the neuroendocrine and the adenocarcinomas. Panel on the left. The panel on the right shows just the adenocarcinomas. So the other figure that I just popped up here on the far right of the slide, though, shows again this frequency that I talked about. It's about an 80-20 split of adenocarcinoma versus 20% neuroendocrine carcinoma in these mice. And that's what we saw in the untreated, the 302 alone, the antibody alone groups. But remarkably, you'll see in the combination group, if you look at that first panel, there are no triangles. If you look at this bar, you'll see that we had 0 neuroendocrine tumors. That either means, quite statistically, so since we should have had 5 or 6, that means that either they're being, immunologically eliminated or they aren't developing in the context of this combination therapy. Either way, this is a pretty interesting finding that we're still hoping to explore clinically. Now, not everybody loves the TRAMP model because of the way that it's generated, because it has this mix of adenocarcinoma and neuroendocrine disease. So we also looked at what is an immunologically extraordinarily challenging model, which is Ron DePino's PB-CRE P10 SMAD purely prostate adenocarcinoma model. Because of the strength of these driver mutations, there are very, very few secondary mutations for the immune system to target in these mice. So immunologically, really nothing has substantial traction here. In our case, actually, with the combination, we did see a pretty significant extension of survival. So, even in this setting, which is immunologically very challenging, we saw a pretty significant benefit to this combination therapy. So all of that was telling us, is really working. We're getting T cells in the tumor. We're seeing these tumors regress, spontaneous tumors, transplantable tumors. Something good is going on here. So we wanted to understand mechanistically how that was working. Now, if we were talking about melanoma, we always talked about CD8 to Treg ratios. The suppressive FOXP3 positive Tregs are keeping your active CD8 and CD4 effector cells from expanding and regressing the tumor. You do see an improvement in these ratios of CD8 to CD8 to Tregs with CTLA-4 PD-1 blockade, with or without TH302, but they're pretty modest changes. You go from about a 1-to-1 ratio to maybe a 3-to-1 ratio. If this were melanoma, that might be 20-to-1, or at least 10-to-1. The reason the CD8 to Treg ratios aren't as deterministic here is that the real suppressive factor holding down these T cell responses is the suppressive myeloid stroma in these cold tumors. And what you can see at baseline, you have this horrible situation immunologically where there are essentially 5 arginase positive MDSC for every single CD8 T cell. That's not a very good position to be in for the adaptive immune response. CTLA-4 PD-1 combination checkpoint blockade increases the T cells a little, but they're still at a tremendous disadvantage. 302 alone, we know, will decrease the MDSC density, but there's still nothing helping the T cells to expand. So you put them together and you get this really synergistic benefit where instead of having 5 MDSC for every T cell, you have 5 CD8 T cells for every MDSC. This is a much better situation immunologically, and we feel that this is really the critical sort of cellular switch explaining the efficacy of this therapy. Now, if we're really removing this heavy suppressive burden on T cells, we should see them look happier in this microenvironment. And that is what we see. We see that their proliferation of both the CD4 T cell effector population as well as the CD8 effectors increases in terms of Ki67. We see that the cytolytic capacity of the CD8s increases both in terms of granzyme B on a per cell basis measured by MFI, as well as in the percentage of the population that's granzyme B positive. So cells are proliferating more, they have higher cytolytic capacity. If, there are ways you can look into the tumor microenvironment and focus in on the subpopulation of CD8s that's tumor specific, as opposed to bystanders, et cetera. Some people use CD39 as a marker for this. Others, based on Rosenberg's findings from the NCI, use a high 4-1BB expressing CD8s as a marker of the antigen-specific population. We chose that approach. When you look at these 4-1BB high CD8s from the TIL, you find that with hypoxia reduction, checkpoint blockade, they're more active in terms of CD44. They make more interferon gamma when restimulated. They make substantially more TNF when we're stimulated. So again, this is telling you not only do we have more T cells, not only do they have greater cytolytic potential, they're more activated and they have greater cytokine effector function. Even in those spontaneous TRAMP mice, which again, we treat, from the time they're 16 weeks old to about 20 weeks old, and then, we pretty much park them for 3 or 4 months until they're 36 weeks old without any further treatment. We were still able, at the endpoint, to observe better CD8 to Treg ratios, better substantially CD4 effector to Treg ratios, and although it wasn't statistically significant, we saw trends of greater CD8 to granulocytic MDSC ratios. And this is, again, with these mice not receiving treatment for 3 or 4 months. So this gave us a pretty good idea that this response, much like in the congenitally marked myeloid progenitor transfer, was pretty durable. So, there was a Tom Gajewski paper that, some people would feel that this is an obvious thing to look at, but someone had to prove it. And really what he showed was that really sometimes whether your anti-tumor T cell response succeeds or fails is really dependent on how well those cells survive when they get to the tumor. If they just get to the tumor and they die at a high frequency, the tumor is going to win and it's going to progress. If they can maintain effector function and survive long enough to actually, get a leg up in the numbers game against the tumor cell, then you have successful anti-tumor immunity. Well, in light of that, I thought this finding was particularly interesting. So if we look at the T cells infiltrating these TRAMP C2 tumors, and we look in the normoxic region versus the hypoxic region, we see very little cell death, so very little caspase positive T cells in the normoxic areas. But if we turn over to the hypoxic areas, we see tremendously high caspase expression in the untreated tumors, where basically almost all the T cells we can find there are at some stage of apoptotic death. Whereas when we reduce hypoxia with combination therapy, we see that sort of, ongoing death process under hypoxia diminished by about half in those T cells. So I think this gets to this longevity standpoint. Now we have T cells that can survive sort of indefinitely in normoxia. We've expanded significantly the percentage of the tumor that is normoxic. And even in these remaining hypoxic pockets, the T cells can survive a lot better than they could before. All right, so all of that data together, really told us that the drug that we found, 302, can really reduce hypoxia, and then our hypothesis that reduction of hypoxia would allow greater infiltration of active T cells and then maybe sensitize some of these cold tumors to checkpoint blockade, had really seemed to hold up, and particularly in some of these really therapeutically intransigent spontaneous tumor models of prostate cancer. We also, some work that I'm not going to show, we also validated the same kind of pattern and finding in head and neck cancer, and actually Greg Dolgoff's group has published some work more recently in head and neck cancer as well. So with all of this data out there, for us, this was enough to say, okay, we should find out if this works in patients. So David Hong led this clinical trial. He's been a fantastic collaborator of mine. He's deputy chair of our phase 1 group, our investigational cancer therapeutics department. And so what we were looking at was in cold tumor patients, those with pancreatic cancer, hormone refractory prostate cancer, as well as HPV negative head and neck, if we treat them just with 302, or its other name is evofosfamide, with ipilimumab, CTLA-4 blockade, can we rescue some of these patients that have failed, many, many lines of prior therapies? And what you can see is we actually had what we felt was a really encouraging result in this early phase trial. It was a dose escalation of ifosfamide. At the 400 dose, the lowest dose, I think we were subtherapeutic. But if you look at where we got to with the higher doses, you'll see that overall across these patients, we had about an 18% objective response rate and over an 80% disease control rate, so meaning stable disease, partial responses, complete responses. And some of these responses were quite profound in very refractory patients. On the right as well, where you see the spider plots, what you can see is, not only are some of these, deeper responses more meaningful, but the stable disease is pretty meaningful in this very advanced cancer patient population. So you see those two lines that run across the middle, kind of yellow colored. Those are pancreatic cancer patients. They had failed, they're metastatic. They had failed all standard of care. They had failed gemabraxane, failed FOLFIRINOX, and were on this trial for over a year without progression. That alone is a remarkable finding, even beyond the actual RECIST level responses. One other thing you'll notice in this graph is the green arrows. So, we were very conservative about the way that we designed this trial up front because we didn't know if, having the drug concurrent with immunotherapy might negatively affect the T cell response. So we only gave the drug ifosfamide in the first 2 cycles along with ipilimumab, and ipilimumab is standard FDA-approved 3 mg per kg 4 doses regimen. What we found though was that patients were coming back to us, 4, 5, 6 months later saying, hey, my CA19-9 is going back up, my PSA is going back up. It looks like my cancer is coming back. Maybe you could give me some more of that stuff you gave me a few months back that actually, got me to where I am now. It was a really good argument, and so we went back to our IRB and put in an amendment so that we could retreat those patients. I know these things take time, and by the time we actually got approval to do this, we were only able to capture maybe 6 or 7 additional patients that we were able to retreat. What you see is almost every patient we retreated responded. So we had a prostate patient that was having a very strong response, that lowest red line. He became nearly a radiographic CR. We had a head and neck patient that was relapsing. We treated her. She stayed on the trial for a couple additional months. Both the pancreatic patients that were on over a year were retreated. In fact, the only patient that didn't really have, an extremely durable response to retreatment was another prostate patient. And unfortunately, he had a grade 3 adverse event to ipilimumab in his last cycle up front. So our IRB would only allow us to retreat him with Evo. Still, he managed to stay on the trial for a couple additional months as well. So the good news about when you retreat patients and they all respond is that it really tells you, this is being driven by this therapy, TH302 plus ipilimumab. It's not some, we had a couple of patients have some miraculous late response to some other therapy they had. That's good because it tells us that our therapy is driving this. The bad news is it probably means we undertreated everybody to some extent if everybody you retreat responds. And we're going to address that in the phase 2. So when I say some of these responses were really profound, and some of these patients were heavily pretreated, I just wanted to give one example. So, this was a metastatic prostate patient, presented with an extremely large involvement of the liver, multiple bone mets. He was hormone refractory. He'd failed, standard taxane salvage chemotherapy. He'd failed a very creative attempt to salvage him with a gemcitabine cisplatin chemotherapy combination that is certainly not standard for prostate cancer, as well as, radiotherapy, all kinds of things. So really, this gentleman had exhausted any available therapies that were out there. Within, our first measurement point at about 8 weeks, he already had very substantial reduction of the liver tumor. You see that here. And once we retreated him, his overall RECIST tumor measurement dropped, almost to minus 90% actually later. And he's still responding. So this was a very durable, very profound response. Some of his bone disease disappeared, et cetera. This was a systemic, durable response against this cancer. So, this was really encouraging. These were really pretty heavily pretreated patients. And so what we wanted to get to was, fortunately, we were able to obtain both blood and biopsies from these patients, and we wanted to see, are we seeing patterns reminiscent of what we saw in our preclinical studies, and what other things do we need to take back to the lab to try to investigate further so we can do a little better, the next round. Now, one thing we saw when we looked at the blood of these patients was that, a lot of the time it's very difficult to look at PBMC or peripheral blood mononuclear cells because the signal to noise is often not very good, and the noise sometimes drowns out your signal. But in this case, we saw a very clear signal that the patients that went on to respond to therapy showed an increase in their peripheral CD8 and CD4 T cell populations that were expanding. So Ki67 positive CD8s and CD4s in the periphery expanded, significantly. It was actually really nice to see statistically significant improvements in such a small trial. Also, on the other side, we saw that patients who came in up front, so now we're talking about a pre-treatment marker of response or non-response in this case, patients that came in up front with a high level of circulating granulocytic MDSC and or monocytic MDSC had less of a likelihood of responding. Now, this was not statistically significant, but it was a pretty strong trend in the non-responders that they had a higher circulating arginase expression in their MDSC. So high arginase PMN MDSC, high arginase monocytic MDSC did not bode well for subsequent response. Now, I mentioned we got biopsies, so we were really interested to see what was going on within the tumor. And obviously, we had blood from all our patients. We only had a limited number of paired biopsies, so 3 non-responding patients and 6 responding patients. Even in this frequency of patients, you could see that there was a really strong trend toward increased proliferation in the CD8 T cells in the tumor, so much like what we saw in the preclinical setting, increased CD4 T cell proliferation. And also, so with pimonidazole, so these patients take pimonidazole, which is the same marker that was marking the hypoxia in all the images I showed you. And with flow cytometry, we could say, how well were the hypoxic areas infiltrated by T cells, by dendritic cells, et cetera. So again, even with the small number of patients, we saw a very statistically significant increase in CD4 T cells able to infiltrate formerly hypoxic areas from which they were probably excluded before. CD8 T cells, similar trend, more infiltration, more hypoxia exposure as they're able to infiltrate and survive in these hypoxic areas. Same with dendritic cells, where dendritic cells are really important for initiating tumor immunity, also very, very excluded from hypoxic areas normally. Finally, we wanted to look at sort of the genetics and say, are we really seeing a clear signature of response versus non-response, which is something else that gives you further confidence that it's your therapy that's driving these responses. I didn't mark out the individual genes here, but what you can see is, between the progressors and the partial responding patients, there's quite a distinct gene signature. That holds up even when we compare the partial responding patients to the patients with very short-term stabilization of disease, less than 2 months. Then even comparing the short-term stable disease to the long-term stable disease patients, we see a very significant difference in gene expression. So this told us we had a very concrete biomarker signature associated with response versus resistance. Now, finally, on the right, there are real limitations to how much you can ask, is hypoxia being reduced, because we're taking biopsies, right? And did those biopsies come from the same place, et cetera, et cetera. There are lots of caveats. Still, we were really interested to see that, if we looked in the non-responders on treatment versus pre-treatment, we saw greater, by transcription factor analysis, we saw greater upregulation of a HIF or hypoxia-induced factor, HIF1 driven transcriptional signature. So hypoxia seemed to be getting worse. Whereas in the responders between pre-treatment and on treatment, we actually saw reduced HIF1 transcription factor analysis signal. So transcriptional patterns associated with HIF were going down in the responding patients, up in the non-responding patients. This says, it's certainly a consistent finding with the mechanism of action of this therapy that we hypothesized. So if we saw this sort of very concrete gene separation or genetic separation of responders versus non-responders, what did we learn from that? Well, if we look at gene set enrichment analysis, hallmark analysis, gene set analysis, what we can see is that the number one gene set associated with non-response or progression was high OXPHOS. And also we see higher fatty acid metabolism, higher glycolysis. This is what we call a hypermetabolic phenotype. And I'll mention that we've observed this before in some settings. So what this represents is these tumors are so efficiently ratcheting up their metabolism that they're essentially starving T cells out for essential energetic nutrients in the microenvironment, and also bathing them in some, rather difficult metabolites for T cells to cope with, lactate, reactive oxygen species, etc. In that context, hypoxia reduction alone may be insufficient to metabolically restore T cell function. On the other hand, in the responders, we saw evidence of underlying, particularly innate inflammatory responses, interferon alpha, inflammatory response genes, etc., STAT5 signaling, as well as some early signs of adaptive immunity in terms of interferon gamma, but not the kind of adaptive immune signature that's been associated with, for instance, response to CTLA-4 or response to PD-1. This is quite distinct. And I think what this represents is when you have some underlying innate inflammation, but the presence of hypoxia is preventing that from actually nucleating a real adaptive immune response that can be mobilized against the tumor, the drug reducing that hypoxia now allows this underlying inflammation to manifest into better adaptive immune infiltration and anti-tumor immunity. So we do have a phase 2 trial planned, approximately 50 pancreatic, prostate, and HPV negative head and neck patients. This is kind of an exciting trial for us. In addition to CTLA-4 blockade, much like you saw in the preclinical data, these patients will also receive PD-1 blockade. Again, we feel like we probably undertreated them a little in the first trial, so they'll be getting evolocumab for the first 3 cycles of their immunotherapy as well. So a little more chance for us to sort of plateau the amount of hypoxia reduction. And we'll be doing some hypoxia imaging of these patients using the 18F FASA PET tracer. So for us, this is a really exciting one. We're hoping it'll open around November or December of this year. Now, where else have we seen this, high OXPHOS signature associated with checkpoint blockade resistance? And I'll say again, Greg Delgoff has published on this phenomenon as well. So we were lucky enough to be involved with another trial, sort of stemming from some work we had done, or very similar to some work we had done preclinically, looking at HPV vaccination and checkpoint blockade, where Bonnie Glisson led a trial at MD Anderson looking at PD-1 blockade with nivolumab combined with the ISO 101 HPV vaccine. And what we saw, if you see on the right panel, was, as opposed to the baseline, let's say, 10 to 20%, more like 13% response rate to PD-1 blockade in these patients, the response rate in this trial was more like 36%. So really, you about tripled the response compared to PD-1 alone. We won't go into that. Vaccination gives us a higher, again, substrate of T cells for that PD-1 blockade to work with. They work together very well. HPV E6 and E7 are ideal antigens because they're virally derived, essential for maintenance of the transformed phenotype. The tumor can't just stop expressing them. And so we were really happy to see this result. But even in this trial, when we looked at what are the gene sets associated with lack of response to this, really therapeutically potent combination of PD-1 blockade and tumor antigen vaccination, OXPHOS, again, number one gene set. And so we were seeing this over and over. So we went back to the lab and we looked at some other data we had from a mouse model. And that data showed us that, there are 2 origins of hypoxia, right, in tumors. So one that we've talked about and the one that we addressed with TH302, disordered vasculature leads to lack of oxygen supply, which, leads to hypoxia. But the other side of the equation is hyperoxygen consumption, so elevated OXPHOS metabolism by tumors, also promotes hypoxia by, basically eating all of the oxygen that's available in the microenvironment. Acting together, these 2 mechanisms establish a truly deep hypoxia that T cells, have, again, a huge struggle to survive and function within. And so, we had done a prior experiment where we took melanoma tumors, so B16 melanoma in mice, treated them with both CTLA-4, PD-1, and PD-L1 blockade in the context of a vaccine. The data I showed you in the very first slide I showed you, which is about 70 or 80% curative. We took the 20% that didn't get cured, put them back into new mice, treated them again, and we kept repeating that until we evolved a tumor line that was completely refractory to the original immunotherapy that cured 80% of them. And then we asked, well, how did these B16 melanomas that were so sensitive to triple checkpoint blockade become completely resistant? And I'll be honest with you, we expected to find they had upregulated new novel checkpoints, or they were making a ton of immunosuppressive cytokine. But the truth is, the main changes were all metabolic. And what you could see is that when we looked on the right panel at this immune-resistant melanoma, we found that it had highly upregulated OXPHOS. There was enhanced adaptation to hypoxia. So this gene set labeled as hypoxia, it's actually a gene set associated with adaptation to hypoxic stress, as well as adaptation to oxidative stress. So when you upregulate OXPHOS metabolism, you're going to be creating as a byproduct a lot more reactive oxygen species. It's the upregulation, the coincident upregulation of the oxidative stress machinery that allows tumors to actually, sort of prosper in the face of this high ROS level. On the other hand, if instead we looked in the microenvironment, so this was a model where we could separate the tumor cells from the surrounding stroma. In the surrounding stroma, as the tumors sort of flourished and increased their OXPHOS and oxygen consumption, the stroma was deprived of OXPHOS metabolism. Basically, the tumor's hyper-consuming all the oxygen. There's nothing left for the stroma. Hypoxic adaptation or adaptation to hypoxic stress was exactly inverted from the tumor. So these stroma were experiencing high signatures of being exposed to high levels of oxidative stress. So think of it as a lack of adaptation to hypoxic stress. And of course, CD8 T cell effector function associated genes bottomed out, right? So you're the immune component of this stroma that can no longer prosecute OXPHOS metabolism and is now being exposed to intense hypoxic and oxidative stress is non-functional. And so we thought of this, mechanistic study in light of finding this hypoxic OXPHOS gene set associated with, lack of response in our hypoxia immunotherapy trial, lack of response in our PD-1 vaccine trial. And so we went back to the lab and said, well, can we also inhibit OXPHOS? What happens if we block that end of the equation at the same time we're trying to remodel vasculature and restore oxygenation with TH302? Now, I will say that, there's some amount of lack of selectivity of OXPHOS inhibitors, and T cells do need some amount of OXPHOS metabolism, particularly when they're activating and expanding initially. However, what we saw with one particular OXPHOS inhibitor shown here, which is the IX-10759 inhibitor, was that in the context of checkpoint blockade, so CTLA-4 and PD-1 blockade, we had a much higher density of T cells in red here when we combined CTLA-4 and PD-1 with 10759. And what you can see sort of in the bar graph is that T cells alone is under this vehicle plus anti-CTLA-4, anti-PD-1. There's pretty much nothing there at day 12, where there's this huge bar. So very significant, both CD3 under normoxia and CD3 under hypoxia with a combination with OXPHOS inhibitor. So that was telling us T cell density in the TRAMC2 tumors was improved in the context of immunotherapy and OXPHOS inhibition. So that led us to investigate a little further, how far we could push this pathway. And we looked at 3 OXPHOS inhibitors. So these are 3 that are sort of clinically relevant. IX-10759, which I showed you, IM-156 from Immunomet, and metformin, which metformin can inhibit OXPHOS at high concentrations. It's a little bit of a dirty drug because, as you all know, it also does many other things. However, this is where we focused our efforts. And that was just seahorse assays showing you that they all really do suppress OXPHOS metabolism. What we found when we looked at 10759 or 156 was that when we use them in combination with TH302, which is the pink line in the graphs on the left, they had a very significant additive benefit, maybe even synergistic, in suppressing TRAMC2 tumor growth. So even without putting immunotherapy on top, just combining TH302 and OXPHOS inhibition, again, which is for us getting to both the sort of push-pull of the origins of hypoxia, had a very strong therapeutic benefit. Now, in the right panels, you see we did combinations with CTLA-4 and PD-1 blockade. And here in the combination setting, the tumors were, essentially wiped out. So we really saw, some very encouraging data. Unfortunately, the tumors were really wiped out both by the combinations of the OXPHOS inhibitor alone and checkpoint blockade, as well as the triple combination with CTLA-4, PD-1, and TH302. So we have follow-on studies in a harder to treat tumor model where we're trying to really get a cleaner idea of how much additive or synergistic benefit there is, where we're not really just curing everything with all the combinations. However, we found this really encouraging, and it's something we're trying to develop as a way to address this resistance that we're seeing in the clinic. When we look mechanistically, I won't go into too much detail, but you can see that when we combine TH302 and the OXPHOS inhibitor 10759, we see high-level CD8 Ki67 proliferation in the tumor, high-level granzyme B production, CD4 T effector proliferation is high. A lot of this is driven by the CTLA-4 PD-1 alone, but we definitely see some benefit in the triple therapeutically. Where is that coming from? Well, one place we see it is in downregulation of arginase production by tumor-associated macrophages. Again, I mentioned the benefit of hypoxia to them in terms of M2 polarization and arginase upregulation. There's also a benefit of OXPHOS metabolism, and MHC2 upregulation is really high in the context of arginase downregulation, something we see associated with really like an M1 phenotypic switch. So we're still developing this data. The last pieces aren't published, but we're seeing a really encouraging trend that if we can block tumor OXPHOS or diminish it in the context of restoring oxygenation with TH302, we create a much more favorable metabolic environment for anti-tumor immunity, particularly driven by T cells. So that's it. I want to thank, in particular, the people in my lab that did this work. Priya is the postdoc who's really led all of this. She's fantastic. Arthur, Krithika, and prior Megan and Medan, Ashvin did some of that work I told you about with the resistant melanoma line and seeing OXPHOS and hypoxic stress upregulation. So again, also thank David Hong and Immunogenesis and Molecular Templates for their work on the clinical trial. And thanks again to LabRoots for giving me this opportunity to present, and I hope you all enjoyed the talk.